Thanks, John, for the nice introduction and the invitation also to join this fantastic symposium. Towards the end of my postdoc time at ISB about 10 years ago, Lee mentioned to me, or actually said to me, very soon you'll be sequencing and analyzing thousands of TB genomes. And knowing Lee's potential for vision, it's maybe not surprising that it's actually exactly what we started to do when I joined MRC at the beginning in London, when I started my group there. And we continue to do that, and today I would like to share some of the insights that we have gained through analyzing a lot of MTB bacterial genomes from clinical isolates from around the world. But before, I would like to give you a brief introduction into tuberculosis, a bit following up from the discussions yesterday. So I'm showing this dramatic picture here on the left, really to illustrate what happens if TB cannot be treated anymore. Rather, when the treatment doesn't work anymore and the patient doesn't respond because the bacteria have evolved extensively drug-resistant tuberculosis. TB is primarily a lung disease. It can affect any organ of the human body, but it's essentially the lung also which functions as the vehicle and the mechanism of transmission through aerosol droplets where the bacteria, known as Microbiome tuberculosis, travel from one patient to the next. Now, MTB is also difficult to work with because it grows very slowly on the one hand, and it's a biosafety level 3 organism, which makes everything complicated, from working in the lab as well as shipping strains around the world. Here are the latest figures of TB today, and some of these were really shown yesterday by Nitin. 10 million new cases every year, 1.8 million deaths every year. Here are the numbers of multi-drug resistant TB, extensively drug-resistant TB. What I really would like to highlight here is the bad news. If you look at the case fatality, right, it's more than 50% of multi-drug resistant TB cases who will actually die. And essentially, that's no different from not treating TB at all. Essentially, if you don't treat TB, roughly 50% of the patients will survive, 50% will die. So TB, as several other bacterial pathogens, have really entered what is known today as the so-called post-antibiotic era. Now, why is it that we're struggling still with TB? How come that this ancient disease is still so highly prevalent around the world? There are many problems with tuberculosis. It's extremely complicated, but some of the main reasons are actually shown here. But, oh, I went ahead of myself, so sorry. That's actually the good news. The good news, if you look at the absolute numbers, and obviously it's a bit of a provocative statement, and you will see at the end of my presentation why that is, but essentially only 5% of global TB is multi-drug resistant. I say global TB, that's important. And that's actually the good news, but as you will see, it's slightly naive if you argue that way. Towards the end of the presentation, I will get back to that. So the problems are many, and the key problems are here. The products that we use to try to control TB are hopelessly outdated. A diagnostic tool, which is more than 100 years old. Most TB patients in the world are still diagnosed with smear microscopy, a tool which routinely misses 50% of the patient. So they go back to the communities, transmit their TB until they're sick enough to be detected. Similar problem with the drugs, 50 years old in average. These are cocktails of drugs, 4 or more drugs that need to be taken for 4, 6 months, 2 years sometimes if you have multi-drug resistant tuberculosis. So big problem there as well. And then the vaccine, BCG, which essentially does not work in terms of protecting against pulmonary TB in adults, which is really also the transmissible form of tuberculosis. There are more problems, and one particular problem is dogmas. TB has a lot of dogma. The field of TB has a lot of dogmas. And one which I encountered when I started to work in tuberculosis about 15 years ago is this one here. M. tuberculosis is a clone, literally. That many famous microbiologists at the time, they were saying these kind of things. And as a consequence of this, most of what we know about the biology and immunology of tuberculosis is really based on one laboratory strain known as H37RV, which was isolated 100 years ago, and since then has been evolving in the lab and in mice, so it hasn't seen in humans for a very long time. And now we know that's actually not true, because if you go around the world and collect a lot of TB strains and sequence them, you actually see that TB, the bacteria, come in different flavors. And we refer to these as the lineages. So different human-adapted lineages. So all of these colored variants here are obligate human pathogens, species Microbacterium tuberculosis, so that we currently know of 7 main lineages. And if you actually look around the world, you see that these lineages are non-randomly distributed. So some are much more widespread than others, and some actually highly restricted to particular geographical regions, for instance, in West Africa, for instance, or East Africa in the case of this orange variant, for reasons we actually don't know. So today we would like to continue focusing on this diversity and really looking at this a bit in 2 different aspects, 2 stories essentially. How does the immune pressure influence the diversity that we see in tuberculosis today? And I would argue that this has actually important implications for the development of vaccines against tuberculosis, on the one hand, and similarly, how does drug pressure impact on the diversity of TB? And I would argue that based on the findings there, the new regimens that we're trying to find, the way we're going to deploy these regimens might actually benefit from some of these insights. So how does the immune system impact on the TB diversity? We know from viruses, many viruses, other bacterial pathogens, and also eukaryotic parasites, that essentially this general model is the rule. So the immune pressure through antibodies, T cells, etc., really force the pathogen to try to escape this immunity through methods or mechanisms such as antigenic variation and epitope variation. So this phenomenon is also known as an evolutionary arms race, right? It's an ongoing process, this fight between the pathogen and the host. And so when the first Illumina sequences became available, that's actually quite some time ago, we thought, oh, can we actually look at this in tuberculosis? Do we see if this also happens in Microbacterium tuberculosis? And so a former postdoc in the group, Iñaki Comas, actually built the first ever Illumina-based phylogeny of M. tuberculosis genomes, 21 at the time, which covered actually quite a reasonable diversity of what we know today. And we also then downloaded what was available at the time in terms of the known T-cell epitopes from the International Epitope Database. So these were experimentally confirmed by others and uploaded there. And to our surprise, what we found is the large majority of these T-cell epitopes were totally conserved. Essentially, no single amino acid variation was found across these 20 genomes in these 466 T-cell epitopes. There was a few which had maybe 1 amino acid difference. So that was totally surprising to us. More recently, another postdoc in the lab repeated this analysis with many more genomes. In the meantime, we also know about more T-cell epitopes. Same picture. Large majority, more than 80% of these T cells totally conserved across now 216 MTB strains. So actually the opposite that what we actually had expected. Even more extremely, if you actually look at the evolutionary conservation of these epitopes, using this DNDS, so the ratio of non-synonymous to synonymous mutations, you see that the epitope-containing domains actually evolutionarily even more conserved than essential genes. Again, essentially the opposite than what we had expected. So since the initial observations, we have been thinking about this, or why that might be. And we have our working hypothesis. I know there are many immunologists in the rooms, and actually be interesting to see whether you agree with this hypothesis. I mentioned that TB is an obligate human pathogen. So on the one hand, that means that TB has no other host. There's no environmental reservoir. It really lives in humans. But it's also obligate pathogens in the sense it has to cause disease to transmit from one patient to the next, which is different from many other pathogens. And we also know that if you look at, for instance, like something like this, that immunopathology plays an important role in TB disease, particularly in terms of building these cavitations, essentially destroying the lung, making these holes, transforming the lung into an aerosol machine, which essentially increases the likelihood of transmission. So perhaps, and that's our working hypothesis, TB really wants to be recognized by the immune system because the immune system helps the destruction of the lung, which then will increase the transmission of tuberculosis. There's some indirect evidence for this hypothesis, in support of this hypothesis, particularly if you look at HIV-TB. It's known that HIV is a big or a very strong risk factor for tuberculosis. But it turns out that HIV co-infected patients have the wrong type of disease, meaning they tend to have extrapulmonary TB more often than pulmonary TB on the one hand. And even if they do have pulmonary TB, they tend to have less cavitation, particularly if their CD4 T cells are low. So that's a meta-analysis from Generalists lab based on the available data, so plotting essentially cavitation versus CD4 T cell count, suggesting that somehow CD4 T cells are implicated in the formation of these cavitation. So assuming for a moment that this hypothesis is true, so this is actually bad news for vaccine development. And why I'm saying that, because most of the vaccine candidates to date are trying to mimic natural immunity to tuberculosis. And if the natural immunity is actually to the benefit of the bacteria, then this is perhaps not a good idea. So the alternative approach would be to turn everything around and say, why don't we start with variable antigens? And perhaps the variable antigens might be more protective than the highly conserved ones. And that's a very first step in this direction that we tried to do together with Joel Ernst and NYU, and then Jane Sutherland at the MRC in the Gambia. And so we started with 200 TB genomes from around the world, identified 7 genes which were particularly highly diverse. That's shown in these black spikes around the TB genome. So 7 genes. We then predicted CD4 and CD8 T cell epitopes, designed peptides, and stimulated those, used those to simulate whole blood in 82 TB patients, HIV negatives, in the Gambia. And yes, indeed, so these are the 7 genes. That's the number of patients who actually responded, somehow recognized these epitopes across these 7 genes. Now, we also did something else. For each epitope, we actually designed 2 different peptides. One which was kind of the wild type, and another one which was a mutant version based on this variation that we had discovered. And if you actually look at and then stimulate the same patient with these 2 variant epitopes, and you actually see that in each of these patients, which are essentially represented here by this line, there's actually a number of them who only recognized the wild-type epitope, and another series of patients who primarily recognize the variant epitope. And so what we take from that is not only are these variable antigens recognized by some TB patients, but the actual mutation that we see has an impact in whether this particular epitope is going to be recognized or not. Now, we don't know what that really means, and I will get back to that in a minute. But to further explore whether the diversity of tuberculosis that we see around the world somehow could be linked to some of these phenomena, we did actually a deeper analysis into one particular lineage of tuberculosis, which is lineage 4, which is the red one, which is essentially the only one which occurs all around the world. From that point of view, it's the most successful type of Mycobacterium tuberculosis. It's the one we really managed to colonize the world. So It turns out that if you look at more genomes, you see that red is not equal red. In fact, you can divide all red strains into at least 10 subgroups, so we call them sublineages. That's work from David Stuckey, a PhD in the lab, and Daniela Britz in a postdoc. But Daniela and David, they then screened almost 3,500 lineage 4 isolates from 100 countries, and then plotted the relative frequency of these 10 variants around the world. So there's a lot of dots here. In the previous graph, these would be all red. Right now, you have these divisions in 10 groups. It's a bit difficult to see any patterns there. So we said, let's plot the frequency by lineage, by country. And if you do that, there's an interesting phenomenon. Out of these 10 subgroups, 4 occur in a very restricted region. And the large numbers here indicate the number of countries that we found these sublineages. For instance, and that's not to say that these are unsuccessful TB variants. For instance, if you go to Uganda, more than 70% of TB in Uganda is caused by this particular genotype. But it just doesn't occur anywhere else, really. And that's in sharp contrast to 3 other sublineages, which essentially occur everywhere. And again, the large numbers there are the number of countries. This particular variant here is also known because it includes the infamous KwaZulu-Natal XDR strains in South Africa. But not all of these are drug-resistant, right? Most of these are actually drug-susceptible. And so here we're trying to propose a concept that perhaps is actually an ecological concept, which is seen in botany, for instance, also, that you actually have specialists and you have generalists. If you think about ecological niches and you take the host immune system as the ecological niche where TB really lives, perhaps the generalists are somehow better in crossing across these different niches compared to the specialists. So how does this link to the epitopes? So we generated, or actually downloaded also, existing genomes from roughly 300 of each of these generalists and 300 of this one Uganda specialist. That's essentially the data we'd had, and repeated the epitope analysis I have been showing before. Again, we saw by far the majority of these epitopes are highly conserved across all of these sublineages. However, there was a subtle but statistically significant overrepresentation of epitopes that actually have at least one amino acid change. It's not a huge signal, but it was significant in the generalists compared to the specialists, perhaps suggesting or supporting this idea that generalists are interacting with more different human immune systems compared to the specialists. So with this, I would like to summarize the first part of my talk. The majority of T-cell epitopes are highly conserved in Microbacterium tuberculosis. That's contrary to most other pathogens. But there are some T-cell epitopes. The epitope variation seems to impact the immune response, and the generalists have a slightly more higher proportion of variable T-cell epitopes. Now again, we don't really know what that means. Is it really an example of immune escape? Or is it actually rather some kind of adaptation to different host populations, or perhaps both? And again, perhaps this might be important for vaccine development. Let me now move to the second part of my presentation, which is about drugs. So how do drugs impact on the diversity of Microbacterium tuberculosis? And here again, there's a dogma, which I encountered when I started to work in TB, which is this one. So drug-resistant bacteria are less fit, less virulent, less transmissible. And at the time that was about 10, 15 years ago, WHO had actually endorsed this dogma. And based on this, suggested that don't waste your time and money treating multi-drug resistant tuberculosis patients because it costs between 100 and 1,000 times more. Yes, they might die, but the problem goes away because they don't transmit, supposedly. So that was, I mean, that's true. So obviously I was completely wrong. And this we know now, because dogma is also wrong. I can say that already now. But nevertheless, we had to try and test it, and that's one of the first things I did when I was at Stanford with Peter, is to revisit this dogma, if you will, by generating lab strains, TB strains, which are resistant to rifampicin. That's one of the first key drugs to treat tuberculosis, with different RNA polymerase beta subunit mutations here. And then we did competition experiments, right? So always the resistant strains against the susceptible ancestor. And if you just look at lab strains, it seems the dogma is true. The fitness here, relative fitness of 1 is the wild type, and all of these had actually statistically reduced fitness compared to the wild type. However, when you looked at clinical strains, there was a different picture. Some clinical strains had no fitness defect. Really, it's more complicated. So at the time, you hypothesized perhaps what might be going on, that yes, initially you have a fitness defect when you go from drug susceptible to drug resistant, but that's not the end of the story, because evolution continues. So you have compensatory evolution, right? So it's additional mutations that come up, which bring the fitness back up, and you remain resistant. So the question was, is that actually true in tuberculosis? And so for this, we had to wait, because that was 10 years ago. We had to wait for Illumina sequencing to be invented, to be able to actually sequence these genomes. The strains were sitting in the freezer. Paired isolates from TB patients who had developed resistance over time. So we can compare the initial isolate and this follow-up isolate. That's one thing we did, to try to look for these compensatory mutations. And the other thing we did was we did experimental evolution with tuberculosis, which takes a long time, because TB grows very slowly. That's why we only transferred every 3 weeks. So you can go on holidays in between, and you can come back and do your next transfer. The bad thing, of course, is that it takes a year before you can, get enough generations. Nevertheless, we did that, and again, we sequenced. And the hope here was that we started with our rifampicin mutants, lab mutants, and the hope was that we would select for compensation during this process. And yes, so in both cases, in the in vivo evolved strains from the patients, as well as in the in vitro evolved strains from the lab strains, we actually found RPOC and RPOA compensatory mutations. So these are other subunits of the RNA polymerase. The red part is where the actual resistance mutations are, and in the blue and the yellow part of the RNA polymerase, that's where these other mutations found. Of course, you say, oh, is that some kind of weird artifact, lab artifact? We had to go back to the field to see whether we would find these mutations in clinical isolates, and that's actually what we did. And that's what the graph, which brings me back to this naive view that MDR-TB is only 5% of TB in the world. Yes, in average, that's true. But if you go to other places, for instance, this area of the world, the former Soviet Union, it's up to 30, 40, sometimes even 50% of new TB, which is multi-drug resistant. So the prediction was that if we looked at several hundred MDR strains from around the world, and then we stratify by either low MDR versus high MDR, these compensatory mutations should be overrepresented in the high MDR regions. In fact, that's exactly what we found. So we had 2 different ways of looking at these compensatory mutations. Some, in one way, were more confident, others were a bit less confident, but either way, we found that these were statistically overrepresented in the high burden, high multi-drug resistant TB burden countries in the world. Now, since then, thousands of TB strains have been sequenced, and these RPOA, RPOC mutations keep popping up. I would like just to end with one dramatic example that maybe brings back to the picture I showed you at the beginning, that even in a rich country like Switzerland, TB treatment can go really, really bad if things are difficult. And the reason I'm showing this because we heard about bedaquiline, this one new drug after 40 years that was developed for tuberculosis, and you can see what can happen. And it also shows how you can actually use genome sequencing to look at what happens within patients. So you can follow TB strains inside patients during treatment. Essentially, that's what we did. So what happened here is there was this refugee from Tibet who came to Switzerland and presented with a hugely resistant strain. So it was resistant to 7 drugs, all first-line drugs, many second-line drugs, even third-line drugs, already resistant from the beginning. So this patient was never treated for tuberculosis. So he was infected with a strain which was already resistant. So much for the fitness cost of extensive drug resistance. By the way, it also had the RPC mutation, suggesting it's really a mature multi-drug resistant strains which managed to infect this patient. So imagine the clinicians here had a problem, because how are you going to treat this patient? Nevertheless, they had to do something. And one thing they did is to actually try to get bedaquiline. And at the time, bedaquiline was only available on a compassionate use. So you had to directly contact the company and say, please, can you send us some bedaquiline? We have this very difficult case to treat. And that's what they did. And so what this shows is really the percentage or the proportion of sequencing reads that we plot over time in serial isolates that came out of this patient. And we can actually follow the evolution of resistance, emergence during this treatment. So here at the beginning, as I said, 7 drugs, 100% of the reads supported the resistance of these 7 drugs. So the treatment worked at the beginning. After 2 years, so here's the dates, right? 2 years of treatment, he was cured. At the time, it was in Zurich. You said it's Switzerland, right? It's not Uzbekistan. Unfortunately, after 5 months, he came back, full-blown TB cavitation, everything. And not only that, by the time he had developed this bedaquiline resistance mutation, 100%. So it was fully resistant to bedaquiline, the new drug, which took how many years and how many millions to develop, just in one patient. This resistance mechanism also confers cross-resistance to clofazimine, which is a very old drug which is used to treat leprosy, and now is repurposed to drug multi-drug-resistant tuberculosis. Of course, at the time, again, something had to be done, so the clinicians tried to treat. Capromycin, an injectable drug, was added. There were some other drugs clinicians were hoping that might still be working. Capromycin resistance started to come up. So we lost capromycin. There's actually a second drug, which is not quite there yet, delaminid, which is being developed. At that time, the clinicians contacted this other company and said, please, 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 can we have delaminid? We really need to do something. That's what they did. Delaminid resistance came up. You see actually 2 different clones separately in the same patient coming up very quickly, one then taking over. 5 years later, the only thing what could be done was surgery. Big part of both lungs had to be removed to reduce the bacterial burden. And luckily, the patient is actually alive, amazingly enough, today. This is really just to illustrate how treatment of this extensively drug-resistant TB can be really, really difficult, and how new drugs can be lost very quickly if we don't deploy them in the right way. And so with this, I would like to end by summarizing the second part. So there's compensant mutations in rifampicin resistance, which are overrepresented in MDR hotspots. These are the dark brown areas of the world. But essentially everywhere where there's a lot of MDR, you see these compensant mutations. They're associated with ongoing transmission, associated with so-called amplification of drug resistance, so they help additional resistance to come up. And so essentially this original naive assumption that fitness of resistant strains is just reduced is essentially naive, because compensation can happen. And so with this, I would like to thank a long list of long-term collaborators, the members of the group, funding agency, and thank you very much for your attention.
Thank you, Sébastien, for that particularly terrifying talk. Lee, here, wait, let me hand this to you.
In the case of the last patient, would the correct way to deal with these drugs be to give them all at once so you had triple drug therapy?
Essentially, that this would have been, in theory, the ideal way. But the problem is that, you see, there was a lag of 3 years. So by the time bedaquiline was kind of available, delaminate was not. It's 2 different companies, so they would not talk to each other, because if there's toxic side effects, who would be to be blamed? So that was one of but essentially, yes. And nowadays, that's essentially what we like to do.
So we have 2 people at the microphones. So let's take these questions, and then please save other questions for the panel discussion. Over here.
Could you comment on TB in livestock and wildlife and resistant strains and their role in global control?
Yes, so that's something which I completely ignored, but you might have seen in some of the trees I showed. It said animal strains or Microbiome bovis. So there are these so-called animal-adapted variant of Microbiome tuberculosis complex, which is a larger group of. Now, as I said, these strains are animal-adapted, so they cause disease primarily in cattle. And cattle are not treated against tuberculosis. They're actually just slaughtered. That's essentially what's done, usually. That's particularly true in the UK, which actually still has a big problem with Microbiome bovis in their cattle production. So it's very unlikely that treatment of animal TB has anything to do with drug resistance in humans, because it's really 2 separate worlds. And bovis is also a very bad pathogen in humans, so it occasionally occurs, but it doesn't transmit. It doesn't cause outbreaks and epidemics in humans. So it's really a separate story altogether.
Yeah, fantastic talk, Sébastien. Can you say something about, or do you know anything about whether these strains have more capacity for persistence, and whether anyone is looking at tolerance, not resistance, tolerance?
No, but it's exactly what we like to do. But nobody's ever looked at this. I mean, the only thing we know about, again, H37RV, right, this magical laboratory strain. That's what most people have been working on. But obviously, the question of whether across the family tree of TB there might be some important differences, that's exactly what we'd like to do at some point, yes.
Terrific. Thank you, Sébastien.